chapter 3, part 1 of this is about the cell, and this is some pretty important grounding information. I know I keep saying that, but it does start in explaining the cell, what the cell does, and much of what you have to learn in 142 goes back to how well you have grasped this information. So it becomes pretty important information. Unfortunately, you don't have a whole lot of context at this time, but it is very interesting and important information. The cell is the smallest functioning unit or the building block of you, so animals and plants. Animal and plant cells are slightly different. We are mostly just studying animal cells here because this is human anatomy. But what we're talking about in this chapter is a generic cell. Not all cells have the parts that we'll be discussing, but most of the parts we're discussing are going to be found in all cells. The idea of where cells come from, they all come from pre-existing cells, whether that be soma cells or even sex cells, all come from pre-existing cells before themselves. And they are this smallest, what I keep saying smallest functional unit, but the reason we name them that way is if you take a cell out of the body, it can still stay alive and perform its function. It really doesn't have a whole lot of point of reference then, and that's why we say it's the smallest unit. Each cell itself has to maintain homeostasis of its own little entity, but homeostasis of the organism, so you and me, is based on all of these cells working together to perform homeostasis among themselves, but also as for the unit, the organism itself. There are two basic types of cells. There's the sex cell and the soma cell, so somatic cell. And the big difference between the two is how much genetic data is found in each one. So you have a half complement of DNA in a sex cell. We call it a haploid. Because the two sex cells are going to get together to form a soma cell, and you have a full complement of DNA in the soma cell. Sex cells have a different process with which they replicate themselves. You learned that in 142, or maybe you already addressed it a little bit in other anatomy classes that you've taken. Soma cells replicate via mitosis. And all soma cells have the same DNA data, but not all of that DNA data that's found in those cells is going to be active in individual cells. So again, we're talking about a generic cell here, anything that's possible in a cell, but this is not true of every cell in your body. There is this thing called differentiation, which we will talk about at the end of this chapter, where these individual units become unique, and that uniqueness turns then into tissue, but that's because certain parts of DNA are turned on in those cells and other parts are not turned on in the cells. I have some pictures that came from your textbook in this particular portion of the lecture that have letters and numbers on them, and I'm going to use those to make it a little clearer what I'm talking about at that time. Hopefully the top of this doesn't get cut off with my in my lecture, but at the top it says extracellular fluid, interstitial fluid. So extracellular, just like the word sounds like, it's the fluid that's outside the cell. So you have fluid outside the cell and inside the cell. And if you're wondering what is that I'm talking about, just think of edema. With edema, you have, that's interstitial fluid, edema. I mean, if you ever have an injury where you have swelling, and that's what we're talking about extracellular fluid or interstitial fluid. So a fluid environment outside and inside the cell. The plasma membrane or the cell membrane, that's an AKA, they can both mean the same thing, separates that cytoplasm, that inside the cell compartment, from what's outside that particular cell. And so you have extracellular fluid generally that is localized around that individual unit that we're talking about, that individual cell we're talking about, but we can't really separate one cell. These cells all operate together for the most part in the idea of a tissue, and then you have that fluid outside. So plasma membrane, the big thing we want to talk about this time, this particular group of information, very important because it's the physical isolation, the barrier between what's going on outside the cell and what's going on inside the cell. That might be a barrier for the cell itself, or it might be a barrier for the cell to make sure that what is going on inside of it doesn't affect the whole body. And think of, for outside the cell, think of maybe a negative pH or a negative temperature or a negative concentration of ions, meaning something that's not beneficial to that cell. Or think about inside the cell if actually that cell has been invaded by a virus or some sort of pathogen, and we want that pathogen to be walled off from other cells. So physical barrier, the membrane does. Also regulates the exchange within the cell, so exchange with the environment. There is an optimal concentration for many things that exist inside the cell. And basically the extracellular fluid is there to provide almost like a grocery store for what the cell needs. It's also a grocery store and I guess a dumpster, you might think both. So the cell gets what it needs from the external environment. It may need amino acids to build more of itself. It may need ions. It may need glucose. It may need oxygen. All of that is going to come in from the extracellular fluid. It may need to dump out its CO2. That's going to be what's dumped outside. It may want to dump out ions that it's exchanging, say sodium for potassium or potassium for sodium. So the membrane allows for exchange, whether that be passive or active exchange, allows for waste to be eliminated, and also if this cell is a specialized cell that's supposed to be making a protein or making an enzyme for the rest of the body to use, it allows for those products that the job it is to make for the body to be released out into the extracellular fluid. Extracellular fluid has a relationship with blood, has a relationship with the lymphatic system. So this cell, via its ability to release something, can release it into the extracellular fluid, then be picked up in the blood, also picked up in the lymphatic system, and then into the blood. So this process allows for there to be communication between this individual cell and systemically the body all over. The makeup of what passes through the membrane is based on what's found outside the membrane and what's inside the membrane. So this membrane is sensitive to the environment. It's sensitive to the level of fluid outside and inside, so fluid compartments have to stay within the optimal level, and then the cell membrane itself has structural support, structural proteins. The structural proteins and also anchoring proteins and other kinds, membrane proteins, I should call them that. That's more of a generic name. The cell membrane is made up of what's known as biphospholipid layer. We call it a biphospholipid layer, so it's two layers of a phosphorus and a lipid tail. The phosphorus tends to like water, so the hydrophilic head or the hydrophilic phosphorus likes water, likes the water approaching it. The hydrophobic fatty tail inside the membrane, and you can see that here. If you look at, I guess the letter J is pointing to the membrane, doesn't like water. So you have water being attracted to the surfaces of the membrane, but not going through the membrane. It's a barrier to various items, ions and water and other compounds that it becomes a barrier for. In terms of the proteins, you have integral proteins and peripheral proteins. Basically, those basic types, and then you have other subgroups, I guess you might say, of proteins. Integral proteins tend to go all the way through the biphospholipid layer. You look at K, you look at C here, you look at E and E, those tend to be integral proteins. You also have peripheral proteins that can be bound to the outside or inside of the membrane. A type of peripheral protein here is I, would be a type of peripheral protein. Peripheral proteins are also identifier proteins. So peripheral proteins can sometimes be moved off the membrane or put on the membrane without actually affecting the whole cell itself, whereas integral proteins can't. You can't take an integral protein out of the membrane and keep the cell alive. With anchoring proteins, these can attach to the outside or inside. If you look at E, the kind of long thin E, it has a little hook around the cytoskeleton. You can think of that as an anchoring protein. And then recognition, again, sorry if it gets cut off, recognition proteins. These are the proteins that are on the surface membrane that identify self and non-self. Enzyme receptors, these are on the outside, and they are unique. Remember, enzymes are catalysts. They tend to have a specificity. And what you have are enzyme receptors that bind to what are known as ligands. And these are things such as insulin is a type of ion or hormone that will bind to these receptors. And once they bind to these receptors, it will actually change the function of the cell inside. It doesn't go inside the cell, but change the function inside. In terms of carrier proteins, just like the word says, it's going to transport something through the membrane as well as channels transport through the membrane. These are important that you kind of get these down in your head, different types of proteins you find in the membrane, because they have they're more fleshed out in their function as you move more about learn more about anatomy. And then on the membrane you have certain carbohydrates. And the thing about carbohydrates is they allow things that are floating by to stick to it. So they create a stickiness. So glycoproteins, lipoproteins, and they can be attached to a protein, but what they do is they kind of hang out there and wave, and will cause something like an ion to be attached or captured in there, so then the cell can bring it into itself. In terms of the membrane carbohydrates. All of these are embedded within this biphospholipid layer. So you have proteins embedded, various different types, and carbohydrates attached to proteins that are embedded in the membrane. And all of this is for the function of the cell. And then finally we have the image that comes from your textbook with everything more or less labeled. And you do have your channel proteins, your integral proteins, you have your glycoproteins, and some of the anchoring proteins, and I don't see on this one a recognizing protein, but you do have those on the surface layer of the membrane that designates that cell as belonging to you as opposed to me. So those cells, those recognizing proteins can be that specific. I will leave this here and we will pick up with the information in the next part.